an implementation study which is funded by ELIXIR, and I'm speaking on behalf of the study. So the study is named FAIRification of Genomic Tracks. I'm from ELIXIR, Norway. Yes, and some other study participants are from the EMBL ABI and from the Supercomputing Center in Barcelona. So before I go into the study itself, I'll say some words about the concept of genomic tracks. I'm sure many of you know about this before, but I'll go briefly through the concepts. So basically, a genomic track is then some data that you can place or locate along the reference genome. The reference genome acts like a coordinate system, and you can add lots of different kinds of data here. It could be genomic data, transcriptomic, or epigenomic data. I'm listing some examples here, but there are plenty more. I will not go into detail here. But these are both then things that happen in the DNA or things that are connected to proteins, for instance. The 3D structure of DNA could be also handled as tracks, virus insertion sites, and others. So currently, there are sort of two main users of genomic tracks. The first and historically most important users were the genome browsers, and especially UCC genome browser, but they invented the term track, as far as I know at least. And then you have the Ensemble genome browser, and there are plenty of other genome browsers. In addition to this, there's also plenty of analysis tools and frameworks that can do analysis on genomic tracks. Yeah, so a bit more about the information content in tracks. So tracks vary quite a bit on the underlying structure of the content. In Norway, we have previously defined 15 different track types, which are variants of points and segments, and you also have track types that can interconnect elements from different places in the genome. And this is, for instance, used for chromatin conformation type of data. And there are also different file formats. Different file formats support different variants of these track types. And also different genome browsers and analysis tools also support different variants of these different track types. So this is just on pure data or informational content level. So this is an old slide. We are thinking of the track Hyperspace as a matrix. I've been doing that for a long time in Norway, and basically you can then add different axes. The main axis would be the base pairs. This is then all the chromosomes or genome put together into a long line. And then you can then add different kind of tracks here, and this is the typical genome browser view. But there's also many different dimensions here. For instance, different types of experiments, different cell types, different genomes. And all of these different dimensions, you will need some metadata in order to locate and combine tracks along in the hyperspace. So a bit about the current situation of how tracks are disseminated. So typically there are different groups, small groups or consortia that generate track data or collect them. For instance, the International Human Epigenome Consortium, or IHEC. You have the FAIRiFying, which is for animal genomes. There's cancer genome and plenty of others. So what then has been done is previously is the generation or the creation of the TrackHub registry, which is collaboration between UCC and Ensemble, or the Embl EBI, more precisely. So basically, the track data generators can provide metadata of the tracks in a TrackHub format, submit that to the TrackHub registry, and then those tracks are imported into both the Ensemble genome browser and the UCC genome browser. So this data flow is very powerful, but it has one main weakness, is that it's mostly focused on getting data to the genome browsers. And the metadata is of lesser importance. Well, in the genome browser, the metadata is of lesser importance than what it would be for analysis tools. So being able to extract metadata from the TrackHub registry would be a very important use case for many users. So some issues and lacking features in the context of FAIR. So as I assume many of you know, FAIR is an acronym standing for findable, accessible, interoperable, and reusable. There is many. I will not go into details here, but these are concepts to use when you want to make your data or metadata freely available and usable by other researchers. And it's also a requirement in many new projects now that you have to create data management plans in order to specify how your data is going to be fair. So in the domain of tracks, there are some issues here. The first issue would be that for findable, you should be able to search and import individual track files across repositories, and also outside large consortia repositories. So currently there are some portals available, but that's typically for just the big collections and not for small collections. It should also be possible to search using formal queries. It's quite good possibilities for free text queries, but it's very hard to get exactly the track that you want. And also, in order to be able to connect your tracks into other existing metadata, there should be global identifiers for the studies, samples, experiments, and the tracks themselves. As to accessible, one should be able to get track files in batch import according to some criteria, and also the data files. There should be guaranteed availability. It isn't now. It happens that tracks that were available previously just disappear, and it might be hard to find them again. And also, there should be availability of binary files and file type conversion tools. That's for the accessible part. For the interoperable part, there is currently a lack of universal metadata fields. So typically, each group defines their specific field or from a specific kind of track data. There are different fields, but there are many universal fields that should go together. Unfortunately, now it should be general across different tracks, but such fields have not been specified. Also, the tracks should be annotating using relevant authority terms so that you can easily find tracks that are connected to different terms and not using different variants of the same concepts. As to reusable, one should have the metadata available together with the track data. And the metadata content should also be consistent, high quality, and versioned. Okay, so I will now go a bit through what we have done in this FAIRification of Genomic Tracks study. So this, as I said, is an ELIXIR implementation study with developers from Norway, from Embl EBI, and from Spain. So this is a bit the current situation. You have the Trackup registry, which then pushes the tracks into an ensemble browser and uses CGN browsers and others. So what we wanted to do in this project was to bridge the gap between the existing browser, genome browser-based system, and then the more analytical users. They could be users of frameworks or command line users. So we used then two frameworks as example, the GZ Hyperbrowser, which is developed by the Norwegian group, and then the Epico, which is developed by the Spanish group. So in order to bridge this gap, we first developed this system called Trackfile, which then supports advanced search capabilities, importing metadata from the Trackup registry, and then providing easy search capabilities to the clients. There's also this metadata format called GSUIT, which we have developed in Norway, which we can use as export, and in addition to JSON formats. And then the client, after getting the search, well, doing the search and getting the search results, the client will typically then look into the search results, find the URLs, and then fetch the actual data itself from the web, basically using HTTP or FTP. The reason why we invented or created the Trackfile software was that the Trackup registry currently has too limited search capabilities, and they wanted something more usable and extendable. So in order for this to go together, we needed to define standard for how the metadata should look. So we have then developed first version of standard called FAIRtracks, which is the JSON schema, and the validator for this. And the validators and the schema itself connects through the ontology lookup service to well, and so the validator checks the ontology lookup service whether the provided ontology values are correct. And also we connect heavily to identifiers.org for the global identifiers for various kinds of metadata, so that you can go from metadata in FAIRtracks format into existing repositories to get more information. So in order to develop this FAIRtracks format standard, we did consider important existing standards, authorities and consumers of track metadata or related metadata. But in contrast to many of these standards, what we have been focused on here is basically end user functionality. So we have not been focused, as many of these have, to make sure that all important metadata is archived properly. That is typically a focus when you have this data management plan. But the FAIRtracks has been developed in order to make this as useful as possible. So it's not metadata storage, it's a metadata exchange standard. So the aim would be to bridge the gap between the track metadata registries or repositories and other interoperability resources and the track consuming tools and frameworks or hack-and-slash scripting or command line access to track data. So as I mentioned, this is a JSON schema. It maps towards different ontologies and makes use of identifiers.org resolvable queries. There's 4 main parts of the FAIRtracks standard. Study has experiments. So there's studies, experiments, samples, and tracks. Yeah. So samples point outward to bioschema. Experiments typically point to the GEO or other kinds of existing data repositories, and so on. So some of the issues that we faced in this project when creating the FAIRtracks standard. One issue is the how to organize these main concepts. So as mentioned, we defined 4 concepts: studies, experiments, samples, and tracks. So these concepts have been defined in accordance to existing metadata models and should be mappable. It's not exactly the same terms, but it should be quite mappable to ISO tools and the others. So next question, which we also struggled quite a bit with, was to define which attributes that one should require. So typical issue is that the tool developers or user, they would demand more rigor than the data providers typically supplies. So if you have looked at existing track metadata repositories, there are, even though they are curated, there's many different variants of spelling and how things are tagged. And that creates problems with users. So the users would have everything perfect, of course. It's hard to do that, so we have to do make some middle ground here. So the balance would be to find just enough metadata to require. These are the most important fields. You can add more if you like, but you don't have to. Some other issues: ontologies. Which ontologies should one select? How can one augment metadata by automated use of ontology relations? We haven't done much into that, but we have discussed it. Regarding identifiers.org, yeah, there are some technical issues. You cannot actually go. You can go from a query, which is the input, and get the URL, but you cannot go the other way around. These are the technical issues. We found, for instance, that EGA, which is the European Genome Archive, their experiments are not registered at identifiers.org, and that, which is something that we needed in our test case. So also another thing that we found out was that currently there are no track identifiers in identifiers.org. You identify the datasets, but the tracks, which is a product of the datasets, they're not identified typically themselves. So one idea would be to propose a track registry service. There's also an existing implementation study in ELIXIR, which is a validation study. We have talked to them a bit, and the question has been how to integrate our solutions with their activities. Yeah, so we ended up doing much of our own validation because of time limitations. So we have created some reference implementation in Python and soon Java. There's the REST API where we can provide JSON documents and get validation results. So in order to do this, we had to extend the standard JSON schema with various novel keywords. So for instance, one keyword is the unique keyword, which is used to validate that there are no duplicate keys across documents, which is a specific requirement. Also, we added support for queries, meaning that this metadata field is supposed to be a query that can be handled by identifier.org. Yeah, I think I lost slide here. So the last part would be also a set of keywords for ontologies. So this is then the JSON validator REST API. A few words about Trackfind. So this is then a Java web application that can be used for searching through metadata and extracting search results. So in our implementation study, we have used the Blueprint data, which is a large European project on generating tracks annotations on mostly blood-related samples. So in Trackfind, you can then select visually various attributes, create search results, and weather results. And there's also a REST API for what? No. So So it supports SQL search and different types of object and relationships. It also handles versioning, and it's also planned to be extended with more curation capabilities. We have added import from the Tracker registry, so we can import data there, and this is also the versioning solutions. Okay, so a few words about possible extension. I have mentioned some of this before. One extension would be to add curation, and we're now adding curation into the Trackfind solution so that you can easily take your metadata and make it into FAIRtracks format, and we will provide help doing that. That's the first extension. Then taking these verified metadata and moving it back into the genome browsers. Another possible extension. I mentioned the idea of providing a track identifier registry so that every track out there can have a specific ID annotated to it, connected to it. And fourth possibility is to actually create some storage for tracks, which is not scattered around the web, but in a central location. Yes. In addition, we have also created some client implementations of Trackfind. So one of these are in the genomic hyperbrowser, and there's another one in the Epico, which is still in development. Yeah. In the hyperbrowser, we have done proof of concept analysis using track data. So in this example, the question is, you have then a set of regions for or variants associated with multiple sclerosis. We downloaded different sites of OpenChromatin for various cell types using the Blueprint data, and ask the question which of these cell types are most relevant to multiple sclerosis. And we get to rankings like this. So this is then using the hyperbrowser statistical analysis platform. Yes. So before the names, I will go into some demonstrations. And, Sorry about that. So first demonstration of the Trackup registry implementation. So there hasn't been that much there, but basically what we have done is to add a FAIRified data set, Trackup, which is then the blueprint data. This is then accessible from the Trackfile software. So if you then fetch this metadata from the API, which they provide, you can look at this as a JSON file. In the existing Trackup format. Injected into that format, we have then the FAIR tracks metadata, starting at this point here. And also, there's a validation from the Trackup registry towards our validation service, and in this case, it actually didn't, it failed. So then going on to the validation. So there's both an REST API and command line tools developed. So this demo shows then the REST API used from the command line. So the first endpoint demonstrated is just a basic information endpoint. And here we can use a specific tool called JQ in order to have a prettier look of the results. So these are then the JSON schemas available in the system. Next, in demo, we'll fetch some of the example files from the FAIRtracks repository, which is on GitHub. It's now cloned. And it's a bit slower. So first, we'll validate a single file from this example, which is then doing actually a web access from command line here using curl with a couple of attributes, but quite straightforward. In this case, this one actually validated, so you can see that validated is set to true. Next case was a bit fast, but then you can actually also provide a set of documents. So using JQ, we can then take a set of JSON files, merge them together, and provide all of them into another endpoint called array. So the next demo is then from the Trackfile system. So on the left-hand side, you then search through the metadata. In this case, looking for H3K4 trimethylation ChIP-seq. Looking at samples here, we can then choose the bone marrow. You generate this SQL query in the middle, and you get results on the left-hand side here. And you can click through the results. There's two export possibilities. One is then the ELIXIR format, and the other one is the JSON format. So GSUIT is then a tabular format. It looks like this. It's not so much readable in this way, but you can paste it into spreadsheets and not in the way he tried there. But in this way. Which makes it quite easily to read in a human way and look at it if you want that. JSON is then a standard format which can be parsed by many downstream tools, and yeah, here's how it would look in JSON. So also you can then take some of these queries, which in this case is a biosample query, open identifiers.org, and you will get access to more information about this specific sample. Yes. So also I wanted to do a live demonstration of the hyperbrowser analysis. So this is then a specific test version of the Genomic Hyperbrowser, which is a Galaxy-based system. I'm now trying to do this multiple sclerosis analysis that I mentioned before. So we have a tool, which is a Trackfind client. This now live calls the Trackfind software in the back end. I can select the blueprint repository and you can select the different attributes here. In this case, I would like to look at the DNA experiments. And I can further narrow down my search results here. For instance, I can look at the different cell types and specify some variants here. Now I can just take in a random sample here, so I'll just select 10 of those. I'll now get a GSUIT file from those, and this is now generated by the Trackfind service. Yeah, so it will look something like this. I can now then run some analysis tools in Hyperbrowser. In this case, I would use the tool determine if these GSUIT tracks are coinciding with the target track. Select the multiple sclerosis track. I select this reference GSUIT. I have to select the reference genome, and I can now run this. There was a bug, which also always happens in the live demo. But fortunately, I have here. I will now get this result page. On the top here, there are cell types which are related to the immune system, and it shows something about that multiple sclerosis is immune-related disease. Okay, so that was the last demonstration. So just back to the end slides here. So these are the study participants. We have a mix of experience from the track production and management including Blueprint and Phantom. There's consortia-based management of track repositories experience there, specifically the IHEC and ICGC. We have experience from track lookup and dissemination, typically through registry, and then the track analysis platforms. And these are the developers and the principal investigators. And my boss, Eivind Håvig, is then the leader also of this implementation study at the University of Oslo. So we have a landing page with links to all the different parts of this study with this URL here. And these are some logos. Yeah, it's open here. So here you can look more into the details. The implementation study is not yet fully finished. It will be finished 1st of December. We will provide manuscripts and the final results. So some of these are not in the final versions yet, but they are quite close. So I think that concludes my talk.